So yeah, as Jenny mentioned, I'm going to give you a talk about the whole body begins as a single cell and the journey of this single cell through our research facilities here at the University of Exeter, which I really enjoy because I love technologies and the technical and the science incorporated with technologies. So, but before I'm going to tell you more about the single cell and the research and technology, please let me give you, I like that because I like to know where I'm coming from and tell people where I'm coming originally from. My heart started to beat in Turkey, so I was born in Turkey, and then I was moved to Germany, modified or established my brain, the full education, and then moved from there to Scotland, then back to Germany, and then back again to Scotland because I loved Scotland. And finally, I'm here, south of UK in Exeter, and enjoy the research environment, but also the nice warm weather, which is close to Turkey rather than in Scotland. But as I told, I really enjoyed my stay in Scotland and also my educations in Germany. And that's, by the way, is my grandpa, and that's me in my little village in Kırşehir. So that's enough from my history. And let's get back to the story why I think that the single cell is quite important. Here at the University of Exeter, we have really a leading research fields in the microbiology, mycology, immune infection, cardiovascular disease, neuroscience, cancer. These are all medical related research fields, and we have really researchers here at the forefront and world leading. But at the same time, I realized when I came here, environmental, marine, zoology, and botanic, amazing researchers here to collaborate and to understand individual cells in a marine environment or botanic environment. So there's a lot that can be done on the single cell level, and we have to understand the single cell level because these are cells of interest, which makes, I will show you why they are important. In the human environment, we have the blood cells, the immune cells, in the natural environment, the amoebas, in the microbiological world, the yeast, bacteria, plankton, and these are really important research cells because they are giving us indications what's happening in our body. The cells in our body flowing, sorry, it's stopped. So the cells in our bodies, they are flowing in the bloodstream here, there, and the bloodstream is not only really responsible for carrying, but also supplying and communicating between the cells, between the connective tissues, the epithelial tissues, muscle tissues, and nervous tissues. And all these tissues are actually made of individual cells. And the researchers, they are all interested also on how we can make these cells, actually the nerve cells, cardiac cells, from stem cells. I'm sure you heard about stem cells, which is a really important research field as well. So how we can do that? And to do that, we need to understand the mechanism inside of the cells, the intracellular system. And intracellular, that's the treasure inside of the cells. We have the nucleus that is really containing the information, the code, how the cells, how the body is made. And of course, the machinery is like a manufacturing unit with all these individual units like the mitochondria and the Golgi's endoplasmatic reticulum that helps us to build our cells and the tissues. This information is actually within the nucleus, the information, how we are coding. It's like a computer with a board computer. The DNA contains all the information, how we are getting, what kind of a color we will have, or what kind of skin we will have, and how long will be our hairs. So the information will be transcribed into this RNA, and the RNA translates this into the protein. That's our central dogma. That's in the molecular biology. Not only in the human world, in the animal world, from one cell getting to this animal, but also in the plant cell, from one cell, from a different development stages, which is the process of multicellular organisms. Here, what you can see, we get the entire plant. So we need to understand these individual cells to understand what's happening within the organism, within the system. And that's actually important, just coming back to the human system, because the cell makes a tissue, which itself goes to the organs, and this again composes the organic systems in order to have really a functional body system. So that means everything has to work in a proper way on the cellular level. In the cellular level, we have this, oops, sorry for that. In the cellular level, we have on the cellular level, within the cells, we have this cell signaling cascade. So the communication is coming from outside, and whatever is happening from outside, it's causing a cascade within the cells. This looks actually like, give me a second, please. This looks actually like, wait, there you go. This looks like this Tokyo subway system. I hope you have seen this before. You can see inside of this map, we have the central system and the trains are coming in. So what will happen if something is going wrong here? That's exactly what will happen then. In the subway system, the trains will crash each other. What will happen in the human world is that we will really not look well, although some people are thinking that we would look well, but things are getting bad in our body, in different systems of our body. We get disease in different systems, like the skeletal systems, the respiratory system, and the muscle systems, or the nervous systems. So that means very important is that we are really understanding the single cell, which is giving rise to the different species, to the different organ system, plants, bugs, plants, human and animals in the marine environment or in the forest environment. So very important aspect, the single cells. Having looked to that, let me come back to my cell. Actually, that's interesting. When I was searching for nice images about cell for many years ago, when I joined the University of Exeter and I wanted to create a postcard for the Center for Cytomics, I found this nice image from Tautvydas Davayanis. It's a painter from Lithuania. It's a really nice guy who gave me the permission to use this. And the title of these two pictures is actually Journey to the Cell Nucleus. On the left side, part 1, on the right side, part 2. And that's really nice. It shows you how beautiful and how colorful is the journey into this nucleus, because there it's happening. There is the central operational part of the cell, the individual cell. Actually, when I searched for that, it reminded me to a starburst of a solar system. Look how similar is that to this one. And the starburst is the birth of a new galaxy system. And it's composed of these planets. To understand the universe, we need to understand the individual components, right? And for this purpose, for example, the astronomers or the physics people, researchers, they are using special technologies like the Hubble microscope, telescopes, or different type of telescopes. We don't need this more expansive, really high sophisticated system. We are not on the macro level. We are in the micro level. We are coming back to the Earth. Here at the Earth, in the University of Exeter, we have research experts and research facilities that really supports the researchers in different research questions. We have a lot of these research facilities. These are centers of excellence that enhance our research and the teaching by pooling this complex technological infrastructure. Not only instrumentation, data management, but also the associated academic trained scientists. These are really specialized experts in the aquatic center, extramagnetic, simulators, and all these core facilities or research facilities are in different campuses of the university. The Streatham campus here in Devon, here in Exeter, Science Park as well in Exeter, St. Luke's also in Exeter, and Penryn in Cornwall. So we have really a lot of research facilities that we can, and all of them are their own experts. So we are quite blessed with a range of excellent research facilities that allows us to support the researchers. On our website, so if you go to the university's page, the main page, and go to the research. You will find a link called Facilities, World-Class Research Facilities. Click on there and you will find detailed information about this. Please go in there and contact the people if you would like to know more about greenhouse, about the imaging, or about the aquatic resource center. And of course, you can contact me and my colleague about the Center for Cytomics as well. But there are much more research facilities as I'm going to show here. So let me come back to the journey of the cell, of this beautiful single cell, so to understand that. To investigate single cells, biological processes, we need model organisms. I remember during my PhD, I used the mouse as a model organism to investigate the transcription factor in a T cell development or in the immune system development. But there are many, for different biological processes. You can use different type of biological model system to, for example, C. elegans, Xenopus, or Sepias or Drosophila melanogaster. And one of the really really important facilities that is housing animal models is our aquatic resource center. I'm really happy to speak about this aquatic resource center because and I was really happy also to visit this because it's really one of the world-class resource center for aquatic animals, for zebrafish, for trouts, and for other type of species. It houses 14 aqua rooms with 3,000 tanks for zebrafishes. It's really allowing the students to analyze, to investigate the effect of to the health or to the reproduction of development in animal models, in the zebrafish, for example. It's a specialized facility that allows to assess the chemicals that impacts the aquatic organisms, but also the environmental impact, the microplastic. I'm sure you heard about that. Microplastic is a big problem at the moment. So if we are getting out from the animals and we would like to see and view the individual cells, the colleagues are moving from these animal facilities and getting into a really high sophisticated bioimaging center, which is led by really experts in their field. It's managed by the experimental officer, Dr. Christian Hacker, and the director of the imaging center is Gero Steinbeck. They are housing really cutting-edge microscope technology, the electron microscope or light microscope. Here's some examples about the fungi Candida albicans. I'm going to show you a video about this later on. About the blastocytes, using specific markers, dyes, and colors, we can visualize different systems of the individual cells here. Neuronal cells, which is developed from the neuronal stem cell. If we want to see more in depth, we go to the trans-electronic microscope that shows us here an example from a Candida glabrata. That's the whole. And if we go deeper down to the nanometer level, we can see the cell wall, plasma membrane, and the nucleus. So that's the imaging the individual cells outside of the organisms. And now we want to know how these cells are interacting and communicating, how the DNA or the RNAs within the nucleus are transcribed, and what kind of DNA and what kind of RNA are in the cells, in the nucleus. That's very important because the cell nucleus is our treasure, it's our really the safe, it's like the storage of our information, which is very important. We need to protect it very well. So we are transcribing this DNA into the cell. The cell is transcribing this into the RNA, which itself is translating the information into a protein. It's like a machinery, that there's a machinery which is really transforming the RNA or translating the information into a protein. So if we understand what's happening on the DNA and RNA level, we can understand if there is a mutation or if we need to repair the entire system. For this purpose, we have a world-class cutting-edge sequencing facility that provides access to the latest sequencing platforms. And this is led by Aaron Jeffries, who is the director of the sequencing facility, and he has a really high-end and really expert of teams like Karen Moore, Paul O'Neill, and so on, all the other colleagues that are really forming the team, which is really leading in their sequencing research. And at the moment, you know that COVID is one of the top topics. So they are also very highly involved in UK with the COVID sequencing. So let me show you on the next slide how a fungal scientist is using the sequencing for their research. And here, I'm sure you heard enough about my voice, so I'm letting one of the leading fungal scientists speak about sequencing.
Our center studies fungi that cause disease in humans, and we focus on the main killers: Aspergillus, Cryptococcus, Pneumocystis, and Candida. The scary thing about these fungi, and not many people appreciate this, is that these fungal infections kill more people globally each year than malaria or breast cancer. What's even scarier is that there are very few antifungal drugs that can be used to treat these life-threatening infections, and the fungi are developing resistance against these drugs. Now, sequencing is a vital tool in our research efforts in the MRC Centre. So what sort of sequencing do? We do genome sequencing, for example, to identify genes that are involved in the emergence of antifungal resistance in these fungal pathogens. We do RNA sequencing to understand the responses of the fungal pathogens to the host during infection. And we also do barcode sequencing, which really strengthens the sort of competitive assays that we do to understand the extent to which particular genes contribute to infection.
So I hope I could give you an idea about how sequencing is really supporting the fungal research at the Medical Research Council for Center for Medical Mycology here at Exeter University. So that brings me finally also to our Center for Research Facility, to our Center for Cytomics, which is relatively new. So let me explain. So we have the research facility for the whole animal, for the single cell visualization, the imaging facility, and also the understanding of the sequencing and the DNA, RNA, we have the experts. Now we at the Center of Cytomics, we are interested on the whole cell, the quantification of the individual single cell, and analyzing not only the cell surface molecules of the cells, but also the intracellular part of the cells. So in the Center of Cytomics, we are using advanced cytometry technology for analyzing live single cells in a high-speed way, really high-speed way. So this includes not only the analysis, but also sorting and isolating the cells. And this is my colleague, Attila Bebisch, who is the backbone of our Center for Cytomics. It's really very good scientist in this field of cytometry. We used to work also in Aberdeen. Then he moved to Cambridge, to the Babraham Institute, to my colleague Rachel Walker over there. And when I came here in November 2019, I really wanted him to come back and work together and build this Center for Cytomics. So where are we located? We are on the Stratham campus. The XCC is here in the Henry Wellcome Biocatalysis Center. And I'm really glad to be in this building. And also my thanks to Jenny, which really fundamentally supported, and it's really the key person who built this Henry Wellcome Biocatalysis building. So here you can see the map of the Southwest, the Exeter campus, and in the Penryn campus. So in Exeter, we have really two campuses, the Stratham and the St. Luke's, and in Penryn, one campus. So we are supporting researchers on 3 campuses. So what is cytometry? The cytometry is the group of methods that used to measure individual cells, the parameters of individual cells, extracellular, but also intracellular, keeping the cell intact at the same time in a quantitative way. And to understand the entire individual cells in a bundle, this is the study of the cytomics. That means we are really want to investigate the cellular system first on the single cell level and combine this information, this knowledge bioinformatically into a functional cell system. So let me show you on the next slide what can be analyzed by flow cytometry. You see on the left side, that's a crowd of people, right? That's a crowd of people that is somewhere in an arena. And now if I would ask you to quantify the people who carries red, who carries blue, who carries the number of the females, the number of different color of the skin, the size at the same time, and so on and so on, it would take ages. But if we are asking the people to move in and passing a gate in a conical shape and passing a laser barrier here, we can easily quantify. Now imagine people are sitting here, one person is quantifying the yellow color, the other person is quantifying the green, and then the other person the size, and so on and so on. In flow cytometry, we are doing all this simultaneously, and we are able then to quantify these individual beautiful cultures one by one at the same time in a high-speed manner. And even if you see someone is hiding within the crowd, it wouldn't, if you just keep it as an image here and you don't let it flow, then you will probably miss it. But in case of a flow, it has not the chance to pass our gate without not being detected. So the flow cytometry is the quantitative cell analysis technology that provides rapid multi-parametric measurement of single cells, particles, in a flow system. That's why the flow, cyto means cell, passing this light barrier, mainly it's laser, many lasers, and metry is the measurement. So we are measuring cellular parameters in a flow system. But we are not creating images, we are creating numbers. And you can be sure that the cells in this flow system are quite happy. So that's really in the reality. And we can measure all these individual different dyes that I have shown you in the imaging slide, the red and the green. We can quantify this in the flow cytometry. How many green? How many red? Once we have analyzed this, we can also isolate this, purify. So we are purifying also cells in a mixture of populations, either individual cells, single cells, or a population of cells. I will show you later an example on that. The traditional flow cytometry measures individual dyes per individual detectors. So red in the red detector, green in the green detector. Meanwhile, and that actually allowed to measure only a view or to investigate only the view of this dye. Only a part of this dye we could investigate. The new fluorescence full spectral cytometry, which is really at the moment the future of cytometry, allows us to measure the entire spectrum of the dye. So not anymore a part of the dye we are measuring, we can also measure the entire spectrum of one particular dye, so that allows us to use more dyes to understand the single cell, to understand the single cells. So the first time, a couple of years ago, my colleague Joanne Lanningham published using 40 color different dyes. So 40 different dyes she used in one analysis, so to understand and to phenotype the different major peripheral blood cells, neutrophils, eosinophils, or basophils, using a full spectral cytometer, and that is shown here, the Cytec Aurora. So that was the first time to deep analyze the immune cells in the blood. So this is the future, but let's come back to the diseases and how we can combine this now from the perspective of a diseases. So if we look to this tumor, we want to understand the tumor is really one of, or the cancer research is one of the main research fields in the world. And we would like to understand what's happening in this tumor, because it's connected with the vascularization, and inside of the tumors is made of many individual cells, and it's probably also there, the cancer stem cell. So why this is important, the cancer research? Because the cancer is still one of the really main disease problem in the world. Every year, we have really around 20 million cases worldwide, and that's really a lot. So there's a big effort of the research community, how we can diagnose cancer, how we can understand the interaction of the cancer. And one aspect is when we have a tumor, the tumor is releasing markers into the peripheral blood. So the primary tumor is releasing kind of a circulating tumor cells in the blood, or DNAs, or little exosomes. There's a huge research going on to identify these individual biomarkers. One of them, and to find them, is really like looking into like a needle in a haystack. So you are searching for this in the blood and to identify just this needle. If we consider the circulating tumor cells, that's just one circulating tumor cells in 10 million blood cells. And that's really hard to find that. But first, we need to know our enemy in the first step. If we know it, we can label it. We can label the circulating tumor cells. We can detect with a specific dye. The dye is labeled to an antibody, and the antibody can bind to the circulating tumor cells. Once we have labeled the circulating tumor cells, we can use this mixture of cells and run this to another technology called imaging flow cytometer in our facility. We have done this, or I have done this at the University of Aberdeen. So the slides that I'm showing you as next are slides that we analyzed at the University of Aberdeen, but we have this technology also here in the University of Exeter. So we labeled this blood with specific antibodies and specific dyes that is recognizing our immune cells and the circulating tumor cells. This machine is producing images, really fast images, 5,000 individual cells per second, but the images are not a really big high resolution, but it's quantitatively enough. So it's enough to get quantitative information out of this. If we do that and we are looking for the tumor cells, which is yellow, you remember, you can nicely identify the circulating tumor cells, the immune cells, which is then labeled with a red dye, and also we found double positive cells. That means red and yellow. And that's interesting because the yellow are the tumor cells, the yellows are the tumors, the circulating tumor cells, and the reds are the immune cells. That means we can see the interaction of the immune cells with the tumor cells. It's an interesting fact. In a normal microscopic environment, this would take ages, but in an imaging or in a flow cytometry with a speed of 10,000 events per second, this will take really only a few minutes. On the next slide, I want to show you how flow cytometry is used together with the sequencing in a cancer research. And this is really a nice animation that I got from my colleague, Kylie Price, Dr. Kylie Price, who's the Chief Technology Officer and the lead of the Youth Green Cytometry facility at the Malagan Institute in New Zealand. She's really one of the world-leading cytometrists. Please let me show you. So in those animations, she's showing the cell analysis of how we can use the flow cytometry sorting and the analysis. So the immune system plays a vital role and keeps us safe. It protects our body from harmful substances. These are made in various organs such as lymph nodes, spleen, bone marrow, gut, lung, and thymus. To study this immune response to a disease, such as lung cancer, for example, we should know how the immune cells are acting on cell-by-cell basis to better understand what's really happening with the immune cells and the cancer cells, which cells are really attacking to the cancer and how the cells are acting. Flow cytometry will allow us to identify the cell types fast and in a multiparametric approach by labeling these samples with different fluorescent dyes. And we can plot them, yellow here, orange there, or double. Once we analyze, we can now identify the specific immune cells. Now we want to isolate them, as I mentioned to you, with the flow sorting technology. So We can nicely isolate this with the flow sorting, which is really extremely fast with 10,000 per second. Once we have isolated them and identified, we can now sequence and map the DNA. We can now get a profile of the genes and to understand what these cells have done in the tumor environment and which genes are up and down. So we can now modify the cells to make it more efficient in fighting the cancer, increasing their immune response by unlocking the cancer killing ability in patients. So once we have modified this, we can put this back to the patient so that the cancers are targeted directly and removed. This is really just one of the many applications that show for flow cytometry and sequencing together. I hope I could show you here with the animation from my colleague how we can fight cancers if we use this technology, cytometry and sequencing. This is already in a praxis here. It's called CAR T-cell therapy. I'm sure you have heard this. So what they have done here, or what they are doing, they are removing the T cells from the patients, modifying these T cells in the lab, growing them, putting it back to the patients, and allowing the T cell, these specific CAR T cells, to target the cancer. This is already in use in the medical environment. That's really very, very important research field. But also in the basic and biological research questions, the research facilities plays a crucial role. On my next slide, and the movie that I would like to show you, and let the colleagues speak about the Galeria melanola, which our colleagues are using to investigate the immune systems. So please, let's listen to our colleagues, and might be much, much nicer voice than my voice today. Okay, there you go.
There is no doubt that many important scientific discoveries would not have been possible without the use of mammalian models such as mice in research laboratories. However, both the ethical implications and experimental cost of using mice means there's a real need for replacement model systems. This is the wax moth, Galeria melanella. And this small insect has the potential to contribute to medical research whilst vastly reducing the need for animal experiments.
That's really an interesting animal model.
Here at the Living Systems Institute at the University of Exeter, and funded by the UK's NC3R Research Council and DSTL, we house a colony of Galeria melanella that we are using to generate novel tools and protocols for use in infection experiments.
This is one of our...
Currently, the main readouts of infection experiments using Galeria are simply measuring whether or not the organism dies and observing the melanization response, where the organism turns black as part of the systemic immune response. The overall goal of my project here at the Galeria Melanella Research Centre is to interrogate the infection model in finer detail. I hope to add more quantitative ways to analyse infection outcomes to maximise meaningful data outputs from smaller numbers of these insects. Over the last 2 years, I've been making use of the Centre for Cytomics here at the university to try to understand how the cells of the Galeria immune system clear infections.
Flow cytometry has significantly changed the landscape of high-throughput cell analysis research in biomedical research, and has become a very popular tool to answer essential bioscience questions in the fields of marine, plant, microbiology, or basic cell development research. So when it comes to the analyzing of single cells or single particles in the lab setting, flow cytometry is widely used and the comprehensive single cell analysis method. Alongside of its unique analysis capabilities, flow cytometry allows the users to enrich and sort specific cells of interest based on the principle of FACS, fluorescence-activated cell sorting. So we are extremely delighted to collaborate with Jenny Campbell from James Wakefield's research group. Her exciting project on characterization of the Galeria immune system demonstrates the capabilities of flow cytometry beyond its traditional application fields. In this study, we have been using nearly all the possible cytometry techniques to understand the Galeria immune system, phenotyping, sorting, and performing imaging flow to quantify phagocytosis, which is the uptake of the bacteria from Galeria immune cells. As technology scientist, my colleague Attila is involved in consulting and designing flow cytometry experiments with researchers in detail to obtain the optimal solution for their research question. Attila has developed with Jenny a workflow to isolate specific RFP-containing Galeria immune cells for downstream applications.
As well as working to develop novel protocols to analyze traditional infection experiments in Galeria, my lab is undertaking pioneering work to genetically engineer the model. This project was driven by PhD student James Pearce, who generated both green fluorescent and red fluorescent animals. This has allowed the study of both infection dynamics and the development of the organism using the facilities in the Bioimaging Center.
The Bioimaging Center is a major research facility supporting researchers like Jenny in biosciences, the Living Systems Institute, and the wider community with advanced imaging techniques for both light as well as electron microscopy. We are equipped with a range of state-of-the-art microscope systems, ranging from laser scanning confocal microscopes, super-resolution wide field systems, as well as transmission and scanning electron microscopes.
The Bioimaging Center offers an array of high-end light microscopes. These provide specialist and complementary imaging modes to address a range of biological questions using different model organisms. Jenny has been making use of our Zeiss LSM880 with AeriScan. It's a very versatile confocal system, providing high speed, sensitivity, and a large field of view. The unique AeriScan array detector significantly increases the resolution and signal-to-noise in comparison to regular confocals.
The unique combination of our Galeria colony here at the Living Systems Institute and the Centers for Cytomics and Bioimaging has enabled us to progress this critical project. Our transgenic Galeria open up this model to be used in further areas of research replacing experiments on mice. We really hope to encourage the wider uptake of Galeria melanella at the University of Exeter, elsewhere in the UK, and around the world.
Well, I hope this gave you an idea how the different research facilities are supporting the researchers here at the University of Exeter, and especially here the Galeria melanella study from Jenny Campbell. It's really using the entire range of the research facilities from us, Cytomics, so analyzing, phenotyping, but also the imaging to visualize the individual structures, but also she's going into the sequencing to understand the transcriptional and the translational level. So that brings me back to my cell nucleus, actually, to this image. I hope I could give you an idea about this beautiful cell, how these cells can be analyzed, and why it's important to understand single cell. That's really like a card tower. An organism is built like a card tower. If one card in the tower is not really working, then the entire system will fall down. The same in our system, in the body system. That's very important that we are understanding the individual cells in the subcellular level, but also on the molecular level. And here at the Center of Cytomics, we are quite ranging under the investigation on the individual cells, on the cell surface, but also intracellular. So finally, I would like to finish my journey with a final animation through our facility. That's our single cells coming to our lab, visiting our lab. at the Stratton campus, in the BioCat building, in the analyzing lab. This is our analyzing lab. The cells are coming from the marine science, marine research here. So we are collaborating with marine scientists, with microbiology scientists, in a bacterial or fungal, but also plant and zoology researcher wants to know the individual cell components. And the biomedical research is important to really optimum investigate the drugs and the development of proper drug effects. So the single cell is really important, that gives rise to the different species, and of course, a quantitative data analysis, which allows us to visualize the data in a really nice way so the researchers can understand. We are isolating the cells and giving it further to downstream applications like the metabolomics, proteomics, or genomics. So our cells is on the tour in the Penryn campus, Sadler's campus, and the Stratton campus, which has been shown first. So all these three campuses are really visited by our single cells. So we are providing, all the research facilities are really providing expertise in their field and also contributing to the publications of the research in the high impact journals. That's from my side for all the research facilities we have here at the University of Exeter. You will find more information about this. Really, there's a lot: mass spectrometry, engineering imaging, or really clean rooms and other, or neuroimaging center facility and others on the medical campus, which are really very, very important. This one here, V-simulators, if I would have more time, I would have shown you some really nice animations from V-simulators. That's, but more efficient to experience the V-simulators on site is more important than on the presentation. I think if you go on the website and click on the V-simulators and contact our colleagues, you can go and visit them. This is really a nice simulation station. So that's the end of my presentation. I would like to thank to my colleagues who provided me the material for this presentation, and also the Wakefield Lab. I thank to Attila, my colleague, the Aquatic Resource Center, the Sequencing Facility, Bioimaging Center, and of course, James Wakefield's lab, especially Jenny Campbell. My special thanks also goes to Kylie Price, who gave me the animation. She's really doing a lot for teaching in cytometry, and it's nice to have colleagues like her. So also, I would like to thank my colleagues, my former colleagues at the University of Aberdeen. They are really a great team of cytometrists. We worked really very efficient to support the colleagues, the researchers at the University of Aberdeen. So by that, please keep calm. So you don't need to wait to reproduce your experiment like Newton is doing here. Just go with the flow. Thanks very much. And yeah, that's it from my side.